Well, welcome everybody. I'm Oliver Fien, Director of the West Coast Metabolomics Center, and it is my great pleasure today to welcome to us John Sobus. He is a physical scientist in EPA's National Exposure Research Laboratory in the Division of Methods and Exposure Methods and Measurements Division. So his area of expertise is environmental and biological monitoring on chemical stressors using both targeted and non-targeted methodologies. His research activities include field sampling, lab analysis of residential and biological media, and analysis of complex data sets using statistical models. So he got his PhD in 2008 in environmental sciences and engineering at the University of North Carolina, Chapel Hill, and his bachelor at a small college in Salisbury University, Maryland, in environmental health sciences. So before he was most of the time at the EPA as a physical scientist, and before a cross-ORD postdoc researcher, including different types of institutions. He will tell us today about the findings from EPA's non-targeted analysis collaboration, or NTACT trial. Welcome, John Sobus.
Thank you so much, Oliver, and thank you for the introduction. Thank you for the opportunity to speak today. As you mentioned, I've been with EPA for a long time. So let me go into sharing screen mode here, make sure everything's working.
All right, floor is yours.
Excellent. Can I get my pointer? No. Okay, so like Oliver was saying, I got my PhD at UNC Chapel Hill with Steve Rappaport. So clearly a lot of his activities in exposomics has influenced what I'm doing these days and influenced my philosophy. But I've been with EPA in one way or another for I think over 15 years now, which is pretty crazy to say. So anyway, I will be talking about a lot of our research activities with non-targeted analysis in our Office of Research and Development over the last 5 or so years. And I'll be focusing mostly on our recent findings from our non-targeted analysis collaborative trial. Let's see if I can minimize this so I can see what I'm doing. Okay, so I wanted to start with the public health drivers for NTA research, because I know many of the folks on the call today are working in the area of metabolomics or other omics disciplines. But in terms of non-targeted analysis and exposomics, these are the real public health drivers. The reality is every single person on this call today is exposed to hundreds or thousands of chemicals every single day by the consumer products you use, by the buildings you inhabit and the vehicles that you drive, by the air that you breathe, by the food and beverages that you consume. There is increased public awareness that we are exposed to a large number of chemicals, and there's also increased public awareness that we haven't done rigorous chemical safety assessments on large numbers of chemicals. So that heightened awareness has led to some regulatory drivers for EPA initiatives. So you likely know that the Toxic Substances Control Act is administered by EPA, and it covers many industrial and commercial chemicals. So TSCA was actually updated back in June of 2016 to explicitly allow risk-based evaluation of both existing and new chemicals. So it kind of sounds like this should have been happening in the first place, but with the Lautenberg amendments in 2016, TSCA is now allowed to explicitly characterize risk for individual chemicals based on both exposure and hazard data. Now, doing a full-blown risk characterization takes a long time, costs a lot of money, and therefore it's not been done on that many chemicals. And as you can see here, there's about 86,000 chemicals that are currently on the TSCA inventory. So there's this challenge that EPA is facing of how to do risk characterization on tens of thousands of compounds that we know to be in existence, let alone the ones that we don't yet know to be in existence. So at this time, EPA's Office of Research and Development was charged with developing what we call new approach methodologies for rapid risk characterization. So what you need to know is non-targeted analysis methods are really key exposure science new approach methodologies. And what I mean by that is traditional monitoring methods that we use in environmental health sciences just can't keep pace with the needs of amended TSCA. NTA methods allow us to rapidly collect lots of exposure data for lots of chemicals simultaneously. My job for the last 5 years or so has been to lead research teams to develop NTA methods and to evaluate NTA methods to understand the state of the science and to really understand whether or not we can use NTA data to inform some type of regulatory decisions. All right, so what's so great about NTA? Well, you probably know, as many of you are analytical chemists, that targeted mass spectrometry-based analytical chemistry has been the backbone of a lot of public health decisions over the last 4 decades or so. With targeted methods, we pick the chemicals of interest, generally very few chemicals, tens, maybe some hundreds of chemicals, and we build a method around those chemicals. We acquire standards. We often do triple quadrupole mass spectrometry. We're trying to be specific, sensitive, robust, and we're trying to get quantitative trace level analysis of these very important chemicals. So the challenge there is we're picking the chemicals that we look at. So we're looking under the lamppost. And at the same time, we're not covering that many chemicals. As I just showed, there's 86,000 chemicals currently in TSCA, and there's many other environmental statutes that cover many other chemicals. And you're all fully aware of the millions of chemicals that are known to exist in various public inventories. So non-targeted is very different than targeted. We don't start with chemicals of interest, we start with samples of interest. This could be environmental media, it could be biological media. We do very basic extraction and cleanup procedures. We analyze those extracts using high resolution mass spectrometry, and we generate hundreds or thousands of molecular features per sample. We use statistical analysis methods to propose chemical formula, and then we try and understand the chemical structures that could be consistent with those chemical formula. Now, I think metabolomics in many cases would stop at the structure elucidation. But if we're going to use NTA or untargeted methods for environmental applications, we can't just stop at structure elucidation. In my world, this is what they define as finding new problems. If we find new chemicals, but we can't say anything about the risk to those chemicals, that raises lots of flags and alarm bells. So we need to extend beyond structure elucidation to semi-quantitation. And if we get semi-quantitative estimates that may indicate some unacceptable level of risk, we need to figure out the source of these chemicals so that we can implement risk mitigation strategies. So this is in a nutshell what NTA is all about. There's tons of different applications for NTA. First, we can rapidly screen for tens of thousands, hundreds of thousands, or more known chemicals using suspect screening. So these are chemicals that are known to be in existence in public inventories, but chemicals for which there's very little exposure data, and therefore we're not in a good position to make any type of decisions about risks to those chemicals. We can also use non-targeted to discover true unknowns, and this is what's been happening at EPA and elsewhere in the discovery of new PFAS compounds. These are chemicals that weren't in any libraries or databases. These were determined and elucidated through high-resolution mass spectrometry. We know that methods develop rapidly over time, so we can just archive MS data and other files and go back in time and uncover historical exposures once our methods are more further developed. And then finally, there are instances, many instances, where we don't even need to do structure elucidation. We can simply look at aggregations of features and use this to identify source fingerprints. So in my world, we may be able to use aggregations of molecular features to say, this is likely a sample from a contaminated site versus this other site that isn't contaminated. And maybe in an instance of Superfund site remediation, this would have a great deal of importance. So about the time that the Lautenberg amendments went into effect, 2016, is when I got involved in doing non-targeted analysis research. And I've kind of shown just a real high-level summary of some of the work that we've been doing kind of on 2 different planes. On the top plane, we have the work we've been doing in data, tools, and informatics. And it's my understanding that Tony Williams presented, I think, fairly recently for this group. So he probably gave you a rundown of some of these activities and some of these tools. But Tony is of ChemSpider fame, obviously, and he came to us about this time, 2015, 2016, and started working on the CompTox chemicals dashboard with a fairly large team of developers and scientists. And I'm guessing you're fairly familiar with the CompTox chemicals dashboard. It currently contains chemical information on, I think, close to 900,000 chemical substances, but it's not just chemistry data, it's hazard data, dose response, phys-chem properties, exposure data, just a wealth of information that is available to the non-targeted analysis community. And this is really our conduit to chemistry space. And this is something that we use in every analysis. But it's not just the chemical substances that's in the dashboard. Tony and his team actually developed MS-ready algorithms. So in the CompTox chemicals dashboard, chemical substances are registered. And these substances can be salts, multi-component mixtures, they can have stereochemistry. The MS-ready algorithms basically take all of those substances and they convert it into a structure form that we would observe on the mass spectrometer. So this desolvates, destereos, removes salts, separates multi-component mixtures. And those structures and all the data about those structures are then available to analytical chemists to use for non-targeted analysis studies. So the MS-ready information is actually available through download. I'll show you how to get to that. And it's also queryable through a batch search option. So this has been a huge benefit to the NTA community. We've also done a bit of work in trying to evaluate and develop things like retention time models that can be used to discriminate amongst different candidate chemicals that could map to a particular molecular formula or a given observed mass. And I'll talk a bit at the end of the presentation. We've done a fair bit of work evaluating some existing in silico tools for predicting MS-MS spectra. At the same time as we've been developing the data tools in informatics, we've been doing lots of different applications. I can't go into any great detail on any of these, but we've looked at things like Brita filters. The federal government is extremely concerned about contaminants of emerging concern in drinking water. So one way to get at that is to use point of use drinking water filters on the tap to collect samples over the course of a month or more, and then to look at what is entrained in those filter samples using non-targeted analysis. And we've done this very effectively and are actually getting ready to scale this up on a nationwide scale for a pilot study. We've also looked at suspect screening in consumer products, and you may or may not be surprised to find that we found many additional chemicals in consumer products than were listed on consumer product ingredient lists and on MSDSs. So there's a lot more in this stuff than you probably would have thought. We've done a lot of NTA work in model organisms. This is just one example where we looked at produced metabolites from different parent compounds in zebrafish that either did or did not have gut microbiota. And then something that's extremely relevant today is using NTA to look at SVOCs in hospital mass to try and discriminate patients with disease versus those that may not have the disease. So a lot of interesting work. We have some really cool studies right now trying to differentiate materials in recycled products versus virgin products. A really cool study looking at xenobiotics in human placenta samples from women with preeclampsia. That hopefully should be going out the door pretty soon. And then all of these data tools and informatics that I talked about before, we're rolling up into a standalone EPA non-targeted analysis web application. This has been slow to develop, but the more that we do these applications down here, the more that we get contacted by people saying, hey, we would like to kind of recreate what you did. How can we do that? So we're trying to build a web application so that folks can replicate our workflows as efficiently as possible. And then the last thing I'm working a lot on is semi-quantitative methods. I've done a lot of reading on this, and I've done a lot of work on this. And the one thing I don't want to be lost is that semi-quant NTA is not only critical for environmental applications, but it's a multi-step process. So we start with media samples, okay? We do extraction and cleanup, and then we have a prepared sample that we analyze on the mass spectrometer. Mass spectrometer yields mass spectrometry data, and this is the step that most people do. They generate concentration estimates that are consistent with the prepared sample. Everyone can still hear me, right?
Yes.
Excellent. I always get nervous about 10 minutes in that I'm talking to myself. So most semi-quant methods that I've seen today will generate a point estimate for a prepared sample. Okay, for our applications at EPA and for other regulatory bodies, that's not going to cut it. Number one, we need a statistical bounding about any given point estimate. So it's not enough just to say, this is what we think the concentration is, and on average, we're within an order of magnitude. We need to say we have 99% confidence that the true concentration level is between this lower bound and this upper bound. Then we can use it for decision making, but it doesn't stop there. We then need to extrapolate back to the initial media concentration estimates. And again, we need to do this in a statistically rigorous manner. So I just want to point out that for NTA applications in a regulatory environment, this has to happen. And this is where I've been spending a great deal of time lately. I don't have time to get into it today, but I think this is what's next to come for NTA. So I've talked just a little bit about what we do at EPA. We've only been at this, at least my group, for about 5 years. Others have been at it for far longer, and there are many different ways of doing this. So this brings me to the state of the science. I'm sure many of you know of or have written different commentaries or viewpoints. I've likely read many of them. I took these two quotes from two fairly recent articles that I thought were really important for today's presentation. So the first is a viewpoint article in ES&T by Ron Heitz and Carl Jobst. They commented that no single analytical technique is suitable for the analysis of all compounds. Successful non-target screening will require the development of multi-platform approaches, facilitated and validated through interlaboratory comparisons. I think pretty much everyone on the call today would agree with this. I certainly agree with this, which is why we launched the ENTACT study back in 2016. The second paper and the second quote I think is critically important. So this was by Megan Guyader out of Chris Higgins' group at Colorado School of Mines. And they commented in their paper that the novelty of non-targeted analysis, particularly its current lack of implementation by regulatory agencies, has prevented the establishment of streamlined quality assurance and quality control procedures. So they're saying here that the fact that the government isn't implementing it is keeping QA/QC procedures from being developed. I think that's backwards. I think the fact that there aren't QA/QC procedures keeps the government from implementing these methods. So that's what I'm all about. I'm trying to figure out the state of the science to define QA/QC procedures so that we can then implement this within regulatory agencies. So this brings me to the fundamental science questions for the research I'm going to talk about today. These are, I think, the key science questions for the research community. When we talk about NTA methods, how variable are the tools and results from lab to lab? Does anybody really know this? Are some methods and workflows just fundamentally better than others? I think most would agree the answer is yes. How does sample complexity affect performance? If you do really well in a biological matrix, would you do equally as well in an environmental matrix? Are they compatible that way? Can we observe or define the chemical space that a given method would cover? And can we do this in a predictive fashion to know how many methods we need to bound a given chemical space? And then finally, do we have the appropriate sensitivity and specificity with the instruments and methods in hand to do environmental applications of NTA? So this is what's been driving the INTACT study from day one. And these hopefully are the questions that I'll try and answer, at least in part today. So I'll mention very briefly, we had a workshop in 2016. I think some of the folks on the call today were invited and presented. This really led to some interesting discussions, which really was the genesis for the INTACT study. And shortly after that meeting in Research Triangle Park, North Carolina, we internally developed a study and started to work with about 30 different collaborators to implement the study. So we wound up developing INTACT in 3 parts, and each part is conducted in several phases. So INTACT Part 1 is going to be the bulk of the presentation today. That's on the left-hand side here. Here we were working in a best case scenario. So these are only synthetic mixtures. So we have something called the ToxCast program, or the Toxicity Forecaster program at EPA, where we maintain a physical library of about 5,000 chemical substances. These chemical substances are used for in vitro bioactivity screening. For the INTACT study, we realized that we could sample from this physical library and we can put these chemicals into a series of synthetic mixtures. For our applications, we decided to take a little more than 1,200 of the highest quality ToxCast chemicals, and we distributed them across 10 different mixtures. And these mixtures had between 100 and 400 chemicals each. We distributed these mixtures basically to anyone that wanted them, so long as we had enough material. So about 30 collaborators at the end of the day said, yes, we'd like to get these. Yes, we would like to participate. The way we proceeded with the study was to ask each laboratory to perform a blinded analysis using the method or methods of their choosing. They would report back to us on standardized templates, their methods and results. We would then individually unveil the list of chemicals to that group, and they would finally perform an unblinded evaluation and submit to us a final report. So pretty much every group has done this step. I will point out that we have 6 contract laboratories that are obligated under contract to do this work, and to kind of help them complete their work, we provided to them multi-well plates of all of the individually plated INTACT substances. So all 6 contractor laboratories, as well as some other laboratories, got all of the 1,269 INTACT chemicals on multi-well plates. So that was part 1, which is kind of, again, best case scenario in prepared synthetic mixtures, and this is in DMSO as a solvent. So for part 2 of INTACT, we wanted to get more into the real sample realm. So we had lots of discussions about this externally and internally. And we finally settled on using 3 reference materials. So one was reference house dust from NIST. This is 2585. Referenced human serum from NIST. I think this is 1957. And then reference silicone wristbands that were made under contract with Oregon State University. So the thinking here was these reference materials have been well characterized using targeted methods, and papers have been published on the compounds found in these materials and the concentrations at which they were found. So we said, if we send these out and ask for a non-targeted analysis, we can easily compare the results from the NTA studies to those of the previous targeted studies. But we wanted to go beyond that. We didn't just want to know how well does it compare. We wanted to know things like matrix effects and extraction efficiency. So we said, how about, in addition to just sending out extracts of these materials, we actually take one of the intact mixtures, spike the materials, and then send extracts of those 4 to 5 materials. So at the end of the day, each laboratory was given the option to get the 10 mixtures, 3 extracts of the reference materials, and 3 extracts of the fortified reference materials. I'd say probably about half of the participating laboratories declined to receive any of the real samples and opted only to look at the synthetic mixtures. So for INTACT Part 3, here is where we went beyond just the 1269 intact chemicals, and we looked at all ToxCast substances in our physical library. At the time, it was 4,600 substances. Here we partnered with various instrument and software vendors and other select laboratories and gave them the full ToxCast suite on a series of multi-well plates for them to go do experimentation to generate MSMS spectra, to then make that available to their customers and for us to make available to the public. We have gotten a number of submissions from our partners on this to date, and I'll bring up Emma and her group have now, I believe, generated 7,000 or so spectra from 1,000 or so of the intact substances, and this has been posted to MassBank. And I think, if my understanding of the numbers is correct, I think this doubles the number of intact chemicals in the public domain for which there are available MSMS reference spectra. So this is a huge accomplishment and a huge thanks to Emma's group. And I think it's a really great example of what can be done through this type of partnership. So those on the call may be interested in actually getting access to the intact chemistry data. I will say we don't have a portal for those lists of substances, specifically because they're supposed to be blinded because people are doing the analyses. But if you're really interested in accessing these chemicals or any other chemicals, I would point you to the CompTox Chemicals dashboard that I'm sure Tony talked about in recent weeks. So this is the main landing page for the dashboard. As you can see, gives you access to 882,000 chemical substances. This is the web address. If this is completely unfamiliar to anyone, there is instructional videos narrated by EPA scientists. I'm guessing this is mostly Tony, which is probably fairly entertaining. But if you have interest in any specific chemicals, you can search them by name, synonym, CAS, Inchi key. You can also search by products and by assays and genes. If you're interested in specific downloads, some of the things I'm going to talk about, I mentioned the MS-ready structures. You can access all of those files through the downloads menu. And then we have a specific lists menu. So from the standpoint of INTACT, anyone interested in getting the list of chemical substances covered in INTACT, for now you'd have to go to this list here. I went to the list page. I just searched for ToxCast. And then the third list down is our EPA chemical inventory for ToxCast. So these are basically 5,231 chemicals that we currently have in our physical ToxCast library. So the INTACT set is a subset of these 5,231 chemicals. So if you click on that link, it'll take you to this page where you have the structures for all 5,231 chemicals. You can pick a chemical, click on it, it'll take you to the landing page for that chemical. Here we have the drawn structure, the Wikipedia entry, intrinsic properties, structural identifiers, linked substances, which is very useful. And then over here on the left, we have chemical properties, fate and transport parameters, hazard data, ADME data, exposure, bioactivity, et cetera. So a wealth of information that if you're unfamiliar with this, I encourage you to check it out. It is, like I said, the backbone of a lot of our NTA work. I'll point out here that on the main menu bar, there's the option for batch search. This is something that we use a ton. So here you have this box where you can enter up to 5,000 identifiers. So you can enter chemical name, CAS, Inchi key, our DSS Tox substance ID, which is the DTX SID. You can search that and pull back all kinds of different chemical identifiers and metadata. If you're doing NTA experiments and you only know the monoisotopic mass or an observed MS-ready formula, you can enter that here and it will pull back all chemical substances in DSS Tox as linked by the MS-ready structures. So this is really your conduit to pulling back all of that chemistry data when you're doing NTA experiments. Okay, enough of the dashboard stuff. So back to the design of the intact mixtures. This is a really, really critical slide because it sets up all of the results that I'm about to present. So we put a tremendous amount of thought into developing these 10 mixtures. You can see here the mixtures were labeled 499 to 508. You can see that the first 3 mixtures had 95 compounds. The next 2 had 185 compounds. The next 2 had 365 compounds. So that was clearly very deliberate so that we could examine the effect of increasing complexity from the standpoint of the number of compounds on NTA performance. So that was kind of complexity test number 1. Complexity test number 2 came from sample 507. So this was only a 95 chemical mixture, but this was a full isobaric set. So these had mass collisions across the board, and we had a ton of isomers in this sample as well. And then finally, we went bananas on sample 508, and we had not only a lower purity mix, but we had tons of mass collisions and isomers in this sample as well. So these two present 2 additional challenges to examine the effect of sample complexity on performance. I will point out that there was a subset of compounds shown here in orange, 90 chemicals that were replicated not only across these 3, but many were actually in sample 507 and 508 as well. So it was about 90 compounds that were replicated up to 5 times. Then you can see this really little blue bar on the bottom. These were 5 compounds that were replicated across the full sample set. So at the end of the day, there's about 95 compounds that were replicated either 3 to 5 times or up to 10 times. And it's this replication in substance spikes that offers us unique means to assess method reproducibility, which I think is absolutely critical if we're talking about using this in a regulatory context. So getting into our results a bit, and all of the stuff that I'm going to show is published, and I'm just kind of skimming the surface of it. But from day one, we knew that we were not only going to coordinate the trial, we were actually going to participate in the trial. So my co-PI, Elin Ulrich, kind of managed, getting the materials out to everyone and coordinating those communications. And I managed, us actually doing the analysis within our laboratory. So we've now used a number of different methods, but for the first paper, we just did LC-QTOF with ESI positive and negative. And you can see on the upper left-hand side here, this is just a bubble plot of all of the features across both ionization modes across all 10 samples. So we had about 1,200 spike substances, and we observed about 26,000 features. So about 20 times more observed features than spike substances. So that was, at the time, alarming. In hindsight, not so much. But clearly, many of these things were not the spike substances themselves, and they were noise, artifacts, contaminants, et cetera. So we took some time, and we developed some procedures and some code, and applied that to the data, and were able to determine that about half, 12,000 or so of the observed features were likely real, and about 14,000, a little more than half, were likely noise or artifacts. And you can see here the ones in yellow are the ones we kept. They tend to be big, and the ones in gray, many of which are very, very little, are the ones that we removed as likely being noise, artifacts, or contaminants. So at the end of the day, we said about 12,000 features were likely real. Well, through lots of processes, we ultimately matched up these real features to the spike substances and found about 1,000 true positives, which left 1,000 real features that could be false positives. They could also be true positives of things that we didn't intentionally put in those mixtures. I don't really know, and I'm not going to know unless we do a whole lot more work. So that brings us to our own evaluation of our performance. If we come down and try and look at the confusion matrix, we've got, was a substance spiked versus was it identified? We can easily do true positives. So the true positive was as high as 65% across the 10 mixtures, which is pretty decent. We can also look at false negatives. So these were things that were spiked that we didn't observe. So this got as low as 35%. Again, it's very easy to interpret. But I don't feel comfortable completing the confusion matrix. Again, I'm not sure that those 11,000 things are false positives. I think some of those may be correct identifications of things that we didn't intentionally put there. And I can't get at that number unless I do a lot more work. And then finally, there's true negatives. So these are things that weren't spiked, that weren't identified. I'm not sure that there's at all an agreed upon way to get at this. And I'm happy to have that discussion today. But for the time being, I don't feel comfortable putting a true negative rate on this, and therefore I can't complete the confusion matrix, and I can't complete all of my method performance statistics. So this is kind of where we left it with our analysis. So that's a high-level review of our work on the mixtures. Next question, who else is working on this study? Well, we have 6 contractor laboratories, again, that are bound under contract to do the work. We have 6 vendors, which not all are actually vendors, but these are institutions that received all of the toxic chemicals for the sake of generating MSMS spectra. And then we have our general participants, some of which got all the samples and some of which only got the mixtures. To date, as a status update, we've received 19 submissions from the blinded portion of the analysis out of 30 labs. And we've received 15 unblinded submissions. So you could basically summarize it to say about half of the people have actually completed the agreed work. All right, now moving into some of the new stuff. how am I processing the data submissions? Number one, and again, I'm happy to have this conversation today. I am not doing a performance evaluation for a laboratory. I'm doing a performance evaluation for individual methods. So if a laboratory does 4 different methods, I'm going to give you performance metrics for each of those 4 different methods. Second point, I've had several laboratories submit to me results where a number of different structures are being submitted as candidates for one feature. I'm not going to deal with that. I need you to give me your number 1 candidate. And if you can't give me your number 1 candidate at the structure level, then give me your best formula estimation. But I've had to negotiate with some labs that submitted large lists of potential chemical candidates per feature. So that's been problematic. Confidence levels have been a really big challenge. Everyone has submitted confidence levels. I'd say most people are using Szymanski et al., but a fair number of people have custom confidence levels, which raises a whole bunch of questions. For the people that have used Szymanski levels, I'd say more than half have misused the Szymanski et al. Levels, which means I don't agree with their scoring levels. So I've had to engage with the laboratories, have discussions, and revise the reported confidence levels after gaining some consensus with the partners of the lab. So once I've kind of cleaned up all the files, which takes quite a bit of time, I take our spike substance lists, and I'll take the MS-ready mass, MS-ready formula, and MS-ready structure and compare that to what's been submitted by the laboratory. So I deem a spiked chemical to be observed if the structure was reported or if the formula was reported when that chemical was not spiked alongside an isomer. If just the mass was reported, I am not considering it observed. I'm saying at this point, there's some evidence to suggest that it could be observed, but I'm not calling it observed. Something is identified if it has an actual structure match. And then finally, something is reproducible if it's correctly identified at least 50% of the time. Now, the reproducible statistic is only relevant for compounds that were spiked more than once and that were identified at least once. So this is confusing, so I'm going to say it again. So the reproducibility metric obviously only applies to things that were spiked more than one time, but it also only applies to things that were correctly identified at least once. So if something was spiked in 2 mixtures and it was correctly identified in one of the 2 mixtures, that compound would be correctly identified, but it would not be reproducibly identified because it was only correctly identified less than or 50% of the time, and it needs to be more than 50% of the time. So this is a totally subjective determination, and it's something that I made unilaterally, and it may be too late to go back now, but it was my best judgment call at the time. Okay, so this is a method comparison. I put this together months ago. So this was based on 7 labs and 12 methods. I think I've done at least 8 labs and 14 methods now, but this should communicate kind of where we are across the different methods. So let's start with the heat map. Here we have the 12 different methods shown on the x-axis and the 1,269 compounds shown on the y-axis. Each cell in the heat map is colored yellow if it was observed and purple if it was not observed. And I've grouped all the ESI positive methods together, all the ESI negative methods together, and then we have a hybrid method, and then we have a GCEI method. This was actually a GC by GC high-res method. And you can see on top, I just ordered them from the highest number of observations to the lowest. So ESI positive got between 42 and 69% observations. ESI negative got between 21 and 39%. And then the other 2 methods were at 48 and 62% respectively. So this is probably no big surprise to everyone on the call, but ESI positive had better coverage than ESI negative. So we know that, but it's still good to see. It's reassuring. What's also interesting here is that when you look across the ESI positive methods, you see these big chunks, right? So this is more than 200 chemicals that were all missed by ESI positive. On the bottom side, there's about 200 chemicals that were all observed by ESI positive methods. So we're seeing really good trends here across similar methods. So that's what we want to see. Same thing's kind of true for the ESI negative, right? Here's a huge chunk that was missed by ESI negative. Here's another huge chunk that was missed by ESI negative. But a lot of the patterns that we see up here look pretty consistent from method to method. So I'm not getting into any hierarchical clustering or any other machine learning aggregation method here, but we are doing that work on the back end. And the things that we're seeing are in fact making sense, which is great. And then no real patterns with the hybrid and the GC method, but you can see some pretty darn good coverage for the GC method here. Last things I'll point out is that if you look across the very top bar, there's only 5% of the ToxCast or the INTACT chemicals that were not observed by any method. And I show some structures here. I am not a chemist. I would not be the best person to suggest why we haven't observed these, but if anyone does have interest in looking at these structures, please feel free to contact me, and I'm happy to give you the information that I have now, and we can kind of talk through some of this stuff. But again, this is some of the stuff that we're looking at on our end as well. And then finally down here on the bottom, less than 1% of all compounds were observed by all 12 methods. And these are really, really small structures, but you can tell that this is actually a multi-component substance, and that structure is actually shared here. So there's really only 3 unique structures that were observed by all 12 methods. So that's a pretty interesting finding. Okay, this is probably the most important slide, so I'm gonna go over this fairly slowly. I have no idea if this is controversial or not. So this is my best cut at doing a method comparison. And I'm doing this across 3 different dimensions. So we have basically a bubble plot again. And the first dimension of the bubble plot is the bubble size. So this is basically saying how much coverage does a given method have? And this is done in percents, right? So if we look at this kind of grayish bubble, it had a coverage of 0.69. So that means 69% of the spiked substances were observed by this method. So this was a LC-ESI positive method done by laboratory 1. So that's metric number 1. Metric number 2 is precision. Precision is the percent of compounds correctly identified divided by the percent of compounds that were observed. So think about it this way. If you observed 100% of the compounds and you correctly identified 90%, you have 90% precision. If you only observed 10% and you correctly identified 9%, you still have 90% precision. So the final precision metric is independent of the number of observed compounds, which I think is really important. The final metric, again, is the most hard to interpret. This is reproducibility. This basically says, of the compounds that were spiked more than once and that were correctly observed at least once, what percentage did you correctly identify more than 50% of the time? So again, it's kind of hard to wrap your head around, but one thing I want to point out is if there were 100 eligible compounds, that means 100 compounds were spiked more than once, and let's say a method got all 100 compounds correct at least once, that method would have its reproducibility metric derived from all 100 chemicals. If another method only got 10 of the 100 correctly identified once, their reproducibility was only based on those 10 chemicals. So again, the reproducibility metric is independent of the number of correctly identified compounds. So it's scalable across labs. All right, so that's my description of the metrics. Let's look at some of these results, which I think is clearly the most fun part. So the first thing that really jumps out here is this green bubble. So this was a GC by GC EI method, high resolution. They had 62% coverage. They had 95% precision and 85% reproducibility. Excellent work. It's really interesting to compare that to this blue bubble here. So this was just a basic GC-EI method, not GC by GC, not high resolution. You can see that their coverage was cut almost in half. They're down to 36%. Their precision dropped to about 85%, and their reproducibility dropped to about 75%. So those 2 metrics are still okay, but you can see the coverage was really diminished by losing the multidimensional GC and the high res capability. On the high end of reproducibility, we had an LC-ESI positive method from lab 4. They had tremendous reproducibility, 95% reproducibility, but their precision was only about 85%. So this means that they didn't necessarily always get everything right, but if they got it right once, they got it right all the time. The flip side of that was this purple bubble. Okay, so this is the hybrid method from lab 6. They had 99% precision. So that means if they observed it, they got it right at least once 99% of the time. But their reproducibility is only about 65%. So that means although they may have gotten it right at least once, they didn't always get it right. So that's a really interesting contrast between the hybrid method from lab 6 and this ESI positive method from lab 4. A couple other interesting ones. You can see we've got this yellow bubble and this light blue bubble. These were both ESI negative methods. You can see the coverage is much smaller at 22%. Other metrics are pretty good. And then the elephant in the room, this lonely gigantic bubble down here, which had excellent coverage at 69%, but extremely poor precision at 7% and extremely poor reproducibility at 7%. So clearly looking at this plot, you want to have a big bubble and you want to be up here in this upper right quadrant. So I have gone back and forth sending results to different laboratories. And I can say I've been a bit dismayed that in sending results to 8 laboratories now, I've really not ever gotten a reply where people have dug into their results. And it dawned on me pretty early on, that's because some of this stuff's pretty heady, it's pretty deep, and people don't really have time to dig into it. So I had to come up with a way to give a visual performance report. So I started thinking about like, SAT scores and GRE scores and going to the doctor and getting a lipid panel and a blood sugar. Those results come back to you in kind of your demographics. So you're looking at your score relative to other people. So I said that would be a really interesting way to kind of report performance here. So I took the same plot, coverage and precision and reproducibility. And I just kind of said, okay, here's everyone else's stuff shown with a black bubble. I'm not really telling you who did what. And then here's your results in red. You had an 88% precision, 77% reproducibility, 36% coverage. Those are your numbers, and here's where you are graphically relative to everyone else. But then from the standpoint of scoring, there's no guarantee that anyone's going to be 100% in any one given category. So your numbers have to be communicated relative to the best in any other category. So we come over here to our performance scores. So you can see for this particular laboratory, and I did this last week and I sent this to the researcher last week, I have yet to hear back. I said, here's your results. Your precision was 88% of the best score. Your reproducibility was 78% of the best score, and your coverage was 30% of the best score. So to date, this is my best way to communicate these results. I'm totally happy to get feedback. Like I said, I've gotten no feedback from any of the participants to date on whether or not they like this strategy. So in addition to the visual results, I give out basically additional files, additional spreadsheets. So one is a really simple performance summary file. So it's just one file per method, and I say, here's the number of correct identifications, and here's the percentage of correct identifications per mixture. Really straightforward. I always offer, and no one has ever said yes, do you want the individual results files? So there would be 10 results files per method, 1 per sample. And these results files basically say, here's the chemicals that were included in that mixture. Did you have a mass match to that chemical? Yes or no? Did you have a formula match? Did you have a compound match? And then I say, here's the confidence level, either as you reported or as we revised based on our discussions. Here's the confidence level that corresponds to your mass, formula, or compound match. So that's a little bit weird to wrap your head around too, but there's many instances where maybe 2 features were observed for a given spike substance, and maybe one was a small isomer peak. They don't necessarily get the same compound score. So if you hit on a chemical twice, I'm gonna report back to you the highest confidence level associated with that hit. And then finally, I think the composite results file are the ones that just make people's heads spin. This is one file per method, and here I list all 1,269 spiked substances. For each substance, I say, this is how many times it was spiked. This is how many times it was spiked alongside an isomer. This is the number of mass hits you got, the number of formula hits, and the number of compound hits. And then I give the decisions. Did you observe it? Yes, no, or undetermined. And again, undetermined would be you observed the mass, but I didn't think that qualified as being an observed compound. Did you correctly identify the compound, yes or no? And did you reproducibly identify the compound? In my mind, I think people should be looking at these results files and trying to understand why they got the results that they did and how they can improve upon it. So hopefully a lot more can be done there. Here's the challenges to date. I mentioned before, some of these groups are submitting multiple chemical candidates per feature. There's not a whole lot I can do with that. So I've been pretty strict that give me your best guess, and that's what I'm going to go with. I also mentioned before the inconsistent and inaccurate use of scoring metrics. I'd say half the groups have come up with their own scoring scale. You know, we're kind of guilty of this too. I don't know if that's good or bad, but it's worthy of discussion. The people that have used the Szymanski et al. Scale, I'd say more than half have used it improperly. There's dramatically inconsistent and inaccurate chemical reporting procedures. I've gotten chemical names, CAS, names and CAS, only ChemSpider IDs, Inchi keys, smiles, DTX SIDs, DTX CIDs. It's different every single time, and sometimes it's even different for the mixtures within a given laboratory. Some labs report feature numbers that are very consistent with the number of spike substances. Others are wildly beyond the number of spike substances. And it's very unclear to me what feature filtering protocols were used. So I think there's a need to think about things like false discovery rates. But again, I'm really reluctant to get into this because I don't know what we can truly call a false positive and what we can call a true positive that was unintentionally spiked. I've mentioned a couple times there's really been limited engagement regarding collaborator follow-up, and it's just because everyone's busy doing a million things, but I think there needs to be additional discussions on whether or not I'm going about this the right way and what this really means. I've mentioned the false positives versus unanticipated true positives. I've also mentioned the issue with true negatives and trying to complete that confusion matrix and report you know, method performance statistics that are dependent on that confusion matrix. And then I finally think the thing that kind of, I wouldn't say keeps me up at night, but I certainly think about it, is this takes me a long time to do. And it takes me a long time because it's just me, because I manage lots of other projects. So this is a slow process, and I'm being as thorough as I can be, but methods are being developed at a very rapid pace. So I want to make sure that whatever we produce when this is all said and done is still highly relevant, given that the methods are going to be way further down the road than they are right now. All right, so I'm almost done. I just wanted to touch on, that was all mixture performance. I just don't want folks to lose sight of the fact that INTACT is more than just those mixtures, right? We have the SRM dust, serum, and wristbands. So this is just a little excerpt from one of our recent publications looking at the INTACT dust samples. So again, we get pushback all over the place of INTACT is just synthetic mixtures. That's not real world samples. Okay, so this is a comparison of best case versus real world. So my colleague, Seth Newton, who has now published this work, said, I'm going to take on the SRM dust experiment, and I'm going to do 4 experiments. First experiment is best case, right? So this is just basically the INTACT synthetic mixture, where we had 365 chemicals spiked into DMSO. This is experiment number 1. Experiment number 2, we said, okay, we'll take that solvent spike, which is the best case, and we'll actually extract the dust matrix and combine it with the synthetic mixture. So here, we anticipate some loss in performance from matrix effects. In experiment number 3, we actually spiked at a high concentration the INTACT mixture in the dust and then did the extract. So now we have the issue of not only the matrix, but the extraction procedure. And then finally, in experiment number 4, we did the same thing. We spiked the dust, but we did it at a lower concentration. So now we have concentration issues, extraction issues, and matrix issues. So this plot just shows that we started with 365 spiked substances. Based on the best case experiment 1 of just solvent spike, we only recovered about 140 of these compounds. So there was a 63% loss in the number of compounds that we could correctly identify with the method that Seth chose. When we compared experiment 1 to experiment 2, we lost another 10% from matrix effects. When we compared experiment 2 to experiment 3, we lost another 5% from extraction effects. And then when we compared experiment 3 to experiment 4, we lost another 9% due to concentration effects. So whereas we started with 365 spiked and we observed, correctly identified about 140 in the best case, we're now down to 50% correctly identified, getting into a real world sample. So that talks about how we dealt with the fortified dust samples. We also wanted to look at the actual results from previous targeted analyses in the SRM dust. So here, Seth did a literature review with some of our other staff members and pulled together a listing of all the chemicals that have been previously reported in various different chemical classes from the SRM dust. And you can see here we've got a fair number of chemicals across 7 different chemical classes. Well, Seth's experiment was using LC-ESI methods, so we didn't think it was appropriate to do a comparison looking at, say, GC results. So here's the number of compounds that have been reported based on just LC-ESI for targeted methods in the SRM dust. 31 PFAS, 3 BFRs, and 9 OPEs. So 43 total chemicals, shown in purple, that have been previously reported through targeted methods. When Seth used non-targeted analysis, found 12 of 31 PFAS, 4 of 9 OPEs, and then an additional 2 phthalates, which was interesting. So slightly less than half of the compounds reported by targeted methods we were able to observe with non-targeted methods. And that's really not terrible. So over here, we can look at the distribution of the 43 chemicals that we would have hoped to have seen. So here, the concentrations in dust span about 5 orders of magnitude. It's pretty darn huge. You can see in blue, these are the subset of those 43 chemicals that we did not observe with NTA. You can see it spans, 3-4 orders of magnitude. And then in green here, these are the ones that we did observe using NTA. So I imagine we did a statistical test on this at some point, but clearly the ones that we did observe are at a higher concentrations than the one that we did not observe. So this means there obviously is some chemical dependency on the compounds that we are able to observe in a real sample. These are not fortified samples. So that then begs the question, okay, when you did fortify the samples, were those environmentally relevant concentrations? Okay, well in yellow, these are the spike concentrations of the fortified intact samples. You can see very much environmentally relevant. They are right on par with those that we observed that are just naturally in SRM 2585. And then going back to Seth's previous experiment where he did higher concentration spikes, you can see that, yeah, we bumped up about an order of magnitude in concentration. So again, it still begs the question, yeah, we have difficulty with NTA when the levels get really, really low. But how do these levels compare to what we might be concerned about from a health risk perspective? Bear with me for a minute because this is challenging. To answer that question, we worked with some colleagues at EPA that work in the ToxCast program. And we basically took all of the intact chemicals and we said, if we look at all of the ToxCast results and we look at the AC50 values, this is the concentration at which a particular bioassay gave 50% response. If we look at all of those bioassay AC50s and we take the 10th percentile of those AC50 values, and then we do toxicokinetic modeling with Monte Carlo sampling, we can figure out in dust the concentration that would be needed for the most sensitive 5% of the population, this is children, to produce a steady state plasma concentration equal to the 10th percentile of the ToxCast AC50 distribution, okay? So that's what's shown here in red. These are model estimates of dust concentrations that would be important from a chemical bioactivation standpoint. And you can see here, there is some overlap on the bottom end of this distribution, but by and large, the chemicals concentrations that would be of interest from a health risk perspective for the most sensitive population far exceed those concentrations that we're experimenting with using NTA. So this just makes the point that yes, NTA is not as good as targeted at extremely low levels, but there's still obviously value in implementing NTA at these environmentally relevant concentrations to understand whether there are risks to some of these chemicals that may have higher thresholds for bioactivity. This is my last slide of substance. I just wanted to point out, I know a lot of folks here are really interested in silico spectral prediction. We did some work, this was led by Alex Chow and Andrew McCachran, working with me and Tony, where we basically took CFMID, an earlier version of CFMID, and we applied it to all of the DSS tox structures. And at the time of this work, there was 765,000 DSS tox substances. And we generated a database of DSS tox, MS2, and silico spectra. So we predicted EI spectra as well as ESI positive and negative spectra at 3 different energies. So we stored this in a database. So then we came back to our intact samples and we said, hey, we've got some really good MS data from DDA runs. We've already run that against a reference library from the vendor and we got our probable structures. Let's go ahead and take that same MS data, run it against our in silico library, develop some methods and try and identify some tentative structures. So the paper is here. It's a fairly dense paper. I tend to be pretty rigorous when we do these things. But the real takeaway point is kind of shown here in this pie chart, that 111 out of 377 intact compounds with good MS2 spectra, 111 were identified as the top hit both with the reference library and with our in silico library. 88 compounds were additionally the top reference library match that were not the top match for the in silico library, but 77 were the top match with the in silico library, but not with the reference library. So this just makes the point that the in silico libraries are extremely valuable, and in this case actually gives us a lot more coverage, mostly because the reference library that we were using from the vendor was very limited in chemical space. So again, a very, very high level summary of this work. If you're interested, I encourage you to check out the paper. So just about done here. So just overall summary of the work. whether you know it or not, you're likely exposed to hundreds or thousands of chemicals every day through lots of different ways. You know, there's now Lautenberg amendments to TSCA. There's other environmental legislation. There's a need to do rapid assessments, at a minimum to prioritize the things that are in need of rigorous assessment. So new approach methodologies are needed to do this prioritization. Clearly, NTA is a major new approach methodology. It's a really promising exposure science tool. But before it can be used to support risk-based decisions, it requires validation. Therefore, at EPA, we're working to develop tools to support NTA studies, to apply these methods and to help identify and prioritize important chemicals based on risk, and to evaluate the state of the science via the INTACT study. What we found from INTACT so far are really good. NTA methods do appear to be suitable for many of the toxic gas chemicals. Only about 5% to date are not observed by any method, and I expect that number to continue to go down. Clearly, multiple methods are going to be required for broad characterization. There is no one size fits all, and interestingly, less than 1% of the INTACT chemicals could be observed using all methods. I come up with this kind of three-tiered approach for performance evaluation, where we're looking at coverage as the ability to observe a compound, precision as the ability to correctly identify those compounds that are observed, and reproducibility as the ability to consistently and correctly identify compounds. So pretty big ranges. Coverage ranges from 22 to 69%. Precision ranges from 7 to 99%, and reproducibility ranges from 7 to 97%. So clearly we have a low outlier in some instances here. So the good news is the upper bound metrics here are really, really solid. I'm very happy to see that. I think the downside is we would hope to see some elevation of some of the lower metrics from some of these individual labs, and hopefully that's something we can work on as a community. And I've just given a couple examples to show that concentration of a chemical, the media in which it's in, and the extraction techniques that are used in the laboratory is going to affect NTA performance, and we need to be thinking about that. And I've also clearly shown that these data are ridiculously valuable for lots of different applications. I've shown you applications evaluating in silico MS prediction tools, but we're actually using this data as the backbone of our semi-quantitative tool development as well. So with that, just want to thank all of the supporters from EPA. We've had a lot of people that have worked with us over the last 4 or 5 years. This probably doesn't even fully capture the folks, and obviously thanks to all the people that have done the analysis as collaborating laboratories and worked with us to make sense of this data. But particularly want to thank Tony Williams and Elin Ulrich, who have really been major players in the study and have just been wonderful colleagues. So with that, I'm happy to take any questions. And yeah, we'll go from here.
Thank you so much, John. That was spectacular, as we kind of knew beforehand, of course. So Tobias Kind has a question. Are MSMS spectra and retention times available? And you say yes, but also for the retention time, like for the individual compounds, for all 1,200 member library.
Are retention time predictions and MSMS predicted spectra available?
Yeah, I think this is. I think Tobias was asking about experimental ones.
So yes. So the retention time. I don't want to get too deep on the retention time stuff. We did develop a machine learning model for predicting retention times. We did a very basic. We did a comparison of kind of a log P based prediction, an ACD Labs vendor software tool prediction versus I think it was an ANN machine learning method, and basically just trying to evaluate the performance of the retention time models. We didn't find that they gave us that much discriminatory power. We are able to filter out some candidates, but to be honest, we didn't find any of those models necessarily scalable, and therefore as we adapted the methods, it wasn't something that we could readily implement. So I will just say, generally speaking, we have been for about 2 years working with a collaborator to implement a scalable solution. So anything that we would give to date in terms of retention time predictions, I don't think would be that useful. In terms of the pre-predicted in silico MSMS spectra, yes. I need to talk to Tony. That's available to anyone as the database. I need to talk to Tony about how best to deliver that. Again, the thinking is, I mean, for everyone on this call, you just want the database. I mean, you can figure out how to hit the database, how to score it. for the general public that's new to NTA, they're not going to be able to do anything with that, which is why we're developing the web application. So it's just load your file, we'll extract the information, we'll hit the database, we'll bring it back, we'll score it, we'll filter out the bad stuff, and we'll give you the result.
Okay.
So yeah, for this audience, we can figure out how to give you access to that database.
Thank you. Elliot Price asks, are EI spectra for GCMS being collected for these types of compounds as well? And available, I guess, also.
So are we talking EI, the in silico EI predictions, or actual EI spectra?
I'm pretty sure that both, you know, that this is about the experimental data.
Experimental? Yes. And again, I'm trying to be really careful about like not calling out collaborators. Yes, one of the vendors has run either the full ENTACT suite, which is 1269, or the full ToxCast suite, or somewhere in between on an EI platform. To the best of my knowledge, that works complete. I do not think they've shared the data with us, but they are obligated to do so. So I will just say we just hired a new staff member, and I put him in charge of starting to rein that in and to make sure that all of these folks signed material transfer agreements, and they are obligated to provide that data to us. So it does exist, and if we don't have it in hand, I think we can get it in hand pretty quickly. Yes,
let us know. We are also interested in this. As you know, we have massbank.us, and we really like spectra. Emma Szymanski asks if your interpretation agrees with hers in terms of, oh my God, spectra already in Mona. Emma, do you want to explain here? Emma, you would need to unmute yourself. Okay, maybe she's not in there.
She doesn't want to talk.
She wonders, okay, so Emma says her ENTACT spectra are already in Mass Bank of North America. So some of them are already in Mona and available to the public.
Okay. Right, so that's, I had mentioned that she, I think, is the first. So we've had maybe 4 groups that have acquired all the spectra, and some of them have loaded it to our group FTP site. We just need to actively do something with it, so that's on us. I think Emma is probably the first group so far to actually load ENTACT spectra into a public repository, and they have done it in MassBank, which has populated in Mona.
Mm-hmm. Alexandros Siskos asks if there's any assessment as to how false the false identification is. Is it a completely irrelevant identification, or is it at least the same kind of compound but wrong ID, or is it just a different isomer of the correct ID? So
right.
Once you're wrong, how wrong are you?
Right, so that's exactly why I provided the results to say, it was all tiered, right? It was like, did you get a correct ID, yes or no? Did you observe it, yes or no? And then for observation, there was criteria for that, right? So it was like, you observed it, in my opinion, if you got the correct formula. So if it was a different isomer and you reported the wrong compound, but you got the formula right, you're getting credit for getting the formula right. If you got the formula wrong, but still observed the mass, I'm not calling it an observed compound, but I'm saying you did see something with that same accurate mass, give or take 10 ppm, right? So yes, there is this kind of tiered system to allow evaluation of how wrong were you. And that's why I said before, it's kind of up to the investigator at that point to look at the data file and say, okay, my coverage was great. You know, we had that one instance with 69% coverage. So they got the formula right for these compounds 69% of the time. But for those compounds for which they got a formula identification, they only got the compound right 7% of the time. So formula ID was great. Compound ID was terrible. So yes, there's absolutely a gradation of performance there.
Mm-hmm. Dr. Shen from my lab asks, could you elaborate on quantification performance, how close it was, how close or different the reported concentrations were among the labs?
No one to date of the 14 methods I looked at have submitted an estimated concentration. All the semi-quant work that we're doing with ENTACT, we're doing on our own with our own data. No laboratories have submitted any type of guess about concentration.
Mm-hmm. Dimitri Abrahamson is asking, he's wondering if you have any estimates for predictive accuracy of the semi-quantification methods. So he published a paper
Yep.
testing 3 machine learning methods.
Yep.
And they were around 0.3 log units, which isn't actually not pretty good.
Right. So what I'll say to that, and Dimitri and I have had conversations about this before. I haven't had an explicit conversation with a regulator in Washington, D.C. But my suspicion is if we say, hey, we found this chemical and we think the concentration is this, and based upon our semi-quant method, on average, we have an accuracy within an order of magnitude, and the bioactivity concentrations, the AC50s, are inaccurate within an order of magnitude. I think they're gonna say to me, this isn't enough information for me to make a decision. And that's why I personally feel that most of the papers that I've seen on semi-quantitation, there is differences in how measures of accuracy are reported, whether it's RMSE or R square or on average within X fold. I think those are good ways to summarize performance. And I think at a minimum, that community should look at having harmonized metrics to say everyone's going to use within X fold of the true value. But you can't just say on average or a median value. And this is where I'm getting to, and this is my charge to the community, and this is something that we're working on. I think for this stuff to be used in decision making, you have to say, for this chemical, for this point concentration, I have 99% confidence that the true concentration is below this upper bound and above this lower bound. And I think if you can get that information, that's when it becomes immediately useful for decision making.
Mm-hmm. I had also myself a question at the very beginning when you thought like these are the most important questions for the scientific community, quality control and whatnot. I wondered what you think about the MQuack consortium. Will that consortium answer some of the questions you have raised today?
Yes, and I'm not as up to speed on MQuack as I should be, but I am aware of it. And let me talk about something that I do know and make the case that like there needs to be better conversation amongst the group leadership. So how many people on this call are aware of BP for NTA? So that's benchmarking and publications for non-targeted analysis. Very few, right? So most people on that group are not really aware of MQuack.
Oh.
You're really doing a lot of the same things. So I'm losing sight of your main point of your initial question.
The question was like, is MQuack going in the right direction?
Right.
There's like 100 people in MQuack or so.
Yeah. So, so yes, everyone's going in the right direction. The problem is there's probably half a dozen groups that are each going in the right direction. So in my opinion, for efficiency's sake, there needs to be kind of a monthly meeting of the leadership between those groups to make sure this stuff is being coordinated, to make sure that there's reasonable duplication is fine. But what you don't want to do is have people working in silos for 3 years. I mean, I'm telling you right now, we're going to have a handful of papers coming out of BP for NTA that are probably going to be a pretty big deal that no one on this call is a part of. And that upsets me because you're really intelligent people and are probably going to really like some stuff and are probably not so much going to like other things. So I'm not in a position, well, I guess I am in a position to influence some of this stuff, but I try and appeal to the leadership to have this kind of cross working group communication as much as possible, but clearly it's not happening as much as it needs to. And that's because people are busy. But and I don't know if Emma's still on this or not, but I mean, I think Emma's a really good example. Like she was active in BP for NTA, and just we get too busy that we can't keep in touch about this stuff anymore. So at a minimum, that's what I would like to see is just a monthly dialogue amongst the leaders of each of those groups to make sure that these activities are being coordinated. But yeah, each group in their own right is doing the correct stuff.
All right. Does anyone else have questions to our speaker today? Unmute yourself.
Yes, I have a question.
Okay.
Oh no, these questions are hard.
Yes, I have a question.
Go ahead.
So the great talk. It's exciting. I didn't know you work on this exciting stuff, so that's great.
Thank you.
The Toxic Control Act, there's a confidential part and a non-confidential part.
Yeah.
What is in the non-confidential part, actually? Sorry, what is in the confidential part? What kind of data is contained there?
I don't know, and I'm probably know 2 or 3 people that could give you a reasonable answer to that. So all I know is the rules allow, I don't want to use the word loophole, but I can't think of a better word, but the laws allow loopholes to where for submissions of new chemicals, some of that information is protected under CBI law, which is confidential business information law, right? So there's only a handful of people that have the appropriate certification in my facility, which has like thousands of people. Most people, I think, in DC have access to that CBI information, but where I work, there's a special CBI room where you have to have the certification and access to the room to get access to that data. So I could probably get you an answer as to what type of data exists, but the reality is you're never going to have access to it. So if you just want to know, there's two or three people I could put you in touch with that could probably have a decent discussion with you about it. I don't know if it's going to change it's not going to change the outcome, but it might be enlightening.
No, it's because on some products you buy in the supermarket, like a spray, like you don't find CAS numbers. It says like it's an undisclosed mixture, and this kind of upsets me because, you know, how can they do that?
Yeah,
So,
I didn't get into UVCBs. Anybody heard of UVCBs? Unknown and variable composition and biological origin. So huge, like 30-35% of materials in Tosca are UVCBs. So these are, you know, palm oil and eucalyptus oil and other things that are AFFF foams, right? That are these composites of chemicals that are very similar in substance. And during submission of UVCBs, there doesn't need to be a very rigid communication of this percentage of that and this percentage of that. It's you know, these types of molecules from this carbon length to this carbon length. And our decision makers have to make decisions on those products, like you say, with that amount of information. So one of the things that they've come to us as ORD for is to use NTA to try and elucidate some of those structures and try and understand kind of the composition of those materials on a structural level and on a kind of component level, weight fraction level.
and one more question. So the United States or the EPA super funds, I think they're running at around 1,300 or 1,800 places across the US, right? Mm-hmm. And it's $1 billion program. So how is it possible that basically the taxpayer has to pay for the cleanup for many years and years to come, while a business can literally go bankrupt and then just go away?
Yeah, I think a lot of us have those questions, and it's probably one of those things that I probably shouldn't answer for fear of getting in trouble, right? But I don't think it's a perfect system. No, I mean, I think there's a lot of screwed up stuff, right? So let me just say that what I'm about and the work that I do, like I said before, I'm not a chemist, right? But I'm doing this work because I think it's necessary. I think there's a huge connection that needs to be made between awesome chemists like yourselves that do this work and people that have to make hard, fast policy decisions. And my fear is, for the sake of a consumer product, you know, remember the vaping stuff that came up a couple years ago, or chemical dispersants, any type of emergency response situation. If we have a point source polluter, right? As NTA or untargeted grows and grows and becomes more utilized, there's going to be the ability of people to come out and say, hey, a decision was made based on this data. And these methods aren't reliable, right? There's no demonstrated QA/QC, and these don't have good quantitative metrics. That's why I am pushing so hard to say the methods exist now that we can find things that we need to be worried about. We can make rapid decisions, but we have to bolster it enough so that those decisions are defensible. So I can't fix policy, I can't close loopholes, but I can try and bolster and communicate the science to a point to where it's as defensible as it can be.
Great, thank you so much.
Are there further questions? Anyone? Awkward silence is expected here at this point. We'll give people the time to think about if they have a question. All right. That is not the case. Usually now we would like to show you around and talk to you one-on-one and whatnot. I'm sorry that it's not possible, but we hope that we will see you in some instance soon.
Absolutely. Absolutely. Well, thank you again for the invitation. This was a lot of fun. And if anyone, I seriously mean this, if anyone has questions about anything, feel free to reach out. But if anyone just hates the way I'm approaching this, please feel free to tell me, because I've gotten virtually no feedback. And it makes me nervous that I'm doing years worth of work, and I don't want to get to the end and have people be like, yeah, why'd you do that? That was kind of dumb. So I'm happy to accept positive or negative feedback. I really look forward to it.
Thank you so much.
You got it.
Congratulations to your work and good luck.
Thank you so much, everybody. Have a good one.
Bye-bye.
Bye-bye.